All right. In this video, I'm going to show you how to do experiment one. And to be honest, a lot of what you learn in experiment one can be applied to the rest of the experiment. So I will also provide a video for experiment two and experiment three and experiment four. But after watching this video, if you feel comfortable doing the rest of them, then by all means, skip the other videos if you want. But that being said, let's get started. So again, to remind you, the whole idea of any experiment is to test a hypothesis. And earlier in this lab, you provided some hypotheses. And the first one was about genetic drift. So you were asking or answering, what change do you think will come to this population of these large goldfish, right? These koi. How do you think they're going to change if there is no natural selection and there's no migration, i.e. genetic drift, and there's no mutations and there's no sexual selection, no natural selection? How do you think they're going to change over 100 generations with basically nothing, no forces, no outside forces forcing any change, just by genetic drift, which is chance itself? To test your hypothesis, which you've already written down your hypothesis. So to test it, we're going to use this website here. Some people can click on it. I can't. I actually have to right click, and I'm going to copy the hyperlink. I'm going to go back here, open a new tab, paste it, and there we go. So this is the website that you're going to use to run your experiments, your simulations. Now you'll notice it starts off with background and tutorial. And if you read the instructions on the lab, I do technically instruct you to read this, and you can if you want. I recommend it. It doesn't hurt. It basically covers a lot of the same stuff I covered in the introduction. It just does it in a different way, which has its advantages because it's good to get the same concept explained in different ways. So read through this if you want, but you don't necessarily need to. The other one is this, the tutorial. You can look at that if you want, but honestly, again, I think you really could skip it because what I'm going to show you in this video basically is a tutorial. And this tutorial that they provide is a tutorial for all the different possibilities, sort of. So some of the information you don't need. And on the flip side, some of the information is not specific enough. So if I were you, like I said, I think I would skip that if you want. You can look at it if you want, or you can not look at it, your choice. But going back to the lab, again, we are talking about experiment one. And like I've already said, the first experiment is about genetic drift. You want to see how this population of koi changes over 100 generations. And to do that, we need to use that simulation and set these parameters, right? These parameters listed right here. And as it says, to begin with, our starting population is 200 koi, and we have equal numbers. So we have about 50% of all the alleles in this gene pool. 50% of them are big R, 50% of them are little r. Also, in addition to that, that's again the allele. For genotypes, then we have about even genotypes. So that would be about 25% homozygous dominant, so big R, 25% homozygous recessive, which is little r, 50% heterozygous, big R, little r. And this makes sense when I show you. But basically, we're starting off at even numbers. And we're going to run this simulation where they exist for 100 generations without gene flow, which is migration, right? Without mutations, without natural selection, without sexual selection, all this. So to do that, again, we go back to this website and we're going to run an experiment. Now, when you click run experiment, it literally does start running the experiment right away. So you need to be prepared to hit the pause button, because before we run the experiment, we need the parameters to be to our liking, so to speak. So here we go. I'm about to hit run experiments, and then I'm going to click the pause button. Run experiments. There we go. Pause. All right. So now I have paused this, right? Let me check one thing. I'll come back to this. Excuse me. Okay. Still making, had to make sure it's still recording. So before we get started, let's explain these parameters. First of all, this stuff up here, you don't have to worry about any of this. All this says is that our population size starts at 200. Obviously, you can change that if you want, but we don't need to. That has nothing to do with our experiment. The carrying capacity, you'll learn about that later, later in the semester, but this is not going to be used in this experiment. So it's just set at 200, and again, you learn about that later. The male to female ratio is half to half, right? So you have the same number of males to females, and for us, that doesn't matter. Mortality rate, so for every one generation, 10 fish die, but also the brood size. So for every one generation, 10 fish are born. So all this is set perfectly as it should be. Here's the part that is more relevant to what we're doing, even though you don't need to change anything. It says the initial big R allele proportion is 50%, meaning 50% of all the alleles in this gene pool here, right? You can see all these fish, and all those fish, all the genes, all the different alleles, 50% of them are big R, therefore 50% of them are small r. Basically saying we're starting even, right? Everything is even to start with. Down here, for evolutionary parameters, that's these things down here. These are the things we're going to adjust later with the other experiments. But again, this experiment that we're running right now is about genetic drift, meaning gene flow or migration shouldn't be affecting anything. There should be no mutation. There should be no natural selection. There should be no sexual selection. So because of that, we just need to make sure all this is set to 0, basically. For some reason, this website automatically defaults to 1 right here, which is not what we want, because when this is defaulted to 1, this means that we have fish coming in. Here's our population in the middle. Migrant R allele prop, right? Set to 1. That means we have fish coming in from other populations, and they are fish carrying the big R allele. And we don't need that. So we're going to click that and set it to 0. Make sure you set that to 0. So here we go. Migration is set to 0, meaning there are no fish leaving, there are no fish coming. Mutation rate, again, everything's set to 0. Nothing is mutating. Genotype relative fitness. Again, everything is equal, 1, 1, 1, meaning there is no advantage to being orange. There's no disadvantage to being white. There's no advantage or disadvantage to being spotted, right? Everything is just equal. And same thing here with assortative mating, which is sexual selection. There is no sexual advantage to being white. There's no sexual advantage to being orange. There's no sexual advantage to being spotted. So now everything is set as we need it. And I know I talked a lot, but again, this is the first of the experiment, so I wanted to explain everything. But in a nutshell, Cliff Notes, basically what you need to do when you open this before you run this first experiment is make sure that this right here is set to 0. Everything else should be set the way you want it. At this point, you're now ready to run the experiment. Now, before we get into that, let me come back to the lab and explain this. So again, we're testing our hypothesis for experiment 1. And basically what we're trying to do, we scroll down here to table 1. Basically what we need to do is fill in this data. And you'll see I had done a different demonstration. So there's information there that doesn't need to be there. Let me get rid of it. We'll start fresh. All right. There we go. All right. The whole purpose of running this experiment, I mean, really the purpose is to test your hypothesis. But the other purpose, as far as grading is concerned, is you need to fill in this table, right? You need to use the data found on this website to fill in this table. The first thing we need is the initial data, right? So before we run the experiment, we need this initial data. So again, to find this information, we're going to get it from this website. And now that everything, as we've said earlier, is set the way we need it, we're going to go over here to our data. And right here, population size is 196. So here we go. We are starting at 196. Done. What's the proportion of big R alleles? So of all the alleles in the gene pool, what percent of them are big R? So we come over here, 0.52, right? 0.52. So basically 52%. And then we ask, all right, well, then what percent of all the alleles in that gene pool are little r? Come over here, little r is 0.48. So 48%, right? 0.48. So those are the allele frequencies, the allele proportions, the allele percentages, however you want to put it. And now let's talk about genotype percentages or proportions or frequencies, right? There's all the different ways you can say it, but it's all the same thing. So let's say how many fish are homozygous recessive. How many of the fish are little r, little r? How many of the fish are white, right? I'm saying the same thing in different ways. Proportion, let's see, little r genotype is 0.24, right? 0.24, which is 24%. All right, then what percentage, what proportion, what frequency, however you want to put it, what portion of all these fish are heterozygote? Big R, little r, right? Which one of them are, how many of them are spotted? Big R, little r. The answer is 49%, or 0.49, which is 49%. Then we ask ourselves, okay, finally, how many of them are homozygous dominant? How many of them are big R, big R? How many of them are orange? So we come here, big R, 0.27, right? Which is 27%. So there we go. We are done collecting data, the data we need before we start the experiment. And I should point out that technically the population size, let me go back here. Remember when we put in these parameters, population size should have been 200, right? But it actually turned out to be 196. The reason being is, remember when I started this experiment, I hit the start experiment button, but then I had to hit the pause button very quickly. So what had happened was, for a split second, that experiment was running. And because of that, the population changed, right? So because of that, even though the initial size should have been 200, it actually ended up being 196. And on that note, technically, big R's to little r's should be 50-50, right? But in all actuality, it was actually 52-48 because, again, I didn't hit the pause button quick enough. Same thing here with the genotypes. Technically, it should be 25% little r, 25% big R, and 50% big R, little r. But again, I didn't hit it quick enough. So this is the data that I'm going to fill in right here. And I'm done. I've done that. So now we need to run the experiment. Excuse me. Let me fix that. Pretend you didn't see it. Now I need to run the experiment and wait for 100 generations and see what all this data is after it's been around for 100 generations. So we come back over here, hit the play button, and you can see it's going pretty slowly. And you can keep it that way if you want. While the experiment is running, you can watch these numbers as they change as you go from generation to generation, and you can see how your population size is changing. And here how, right now we got 54% big R's and 47% little r's. And you can see how the genotypes are changing as far as percentages. And you can watch that the whole way through if you want. And these are the numbers. And this stuff here is the same data that's here, except it's in a graph, right? There's the population size over time. There's your allele proportion over time and your genotypes. So again, you can just sit here and watch if you want. But remember, the idea here is for you to hit 100 generations. So if you just sit here this long, it'll take a while. My suggestion is you hit this fast forward button until you hit 16 here, because 16 is the fastest speed. So now it'll be much quicker because, again, you're trying to hit 100. And still, as you're waiting for it to hit 100, you can still watch the population size change and the allele proportion change. Same here, right? So homozygous recessive, that represents the white fish. Homozygous dominant, that represents the orange fish. Heterozygous, that's the spotted fish. And you can see how the percentage changes, right? and then you pause it right when you hit 100. And let me say this, 100 is what you're shooting for. Let me go ahead and do this to make sure. Let's assume I messed up. All right, there we go. Let's assume I hit 101 instead of 100. That's okay. That's okay. What you do need to do then is go up here in your graph and actually change it, because it's not after 100 generations. Technically, that's after 101. And that being said, if you wanted to, you could keep running the simulation longer. You don't have to do just 100. You can have a higher number if you want. But you do need to shoot for a minimum of 100. So shoot for that. Like I said, if your number's a little bit off, that's okay. Just make sure you reflect it right here. Put your actual numbers of generations. Anyway, so now we're done with the simulation. It is now time to put this data in, just like we did before. But hopefully the data has changed because we've gone through 100 generations. So we come back here. Population size is now 199. All right, population size after 101 generations is 199. All right, what's the proportion of big R alleles? Come back over here, proportion of big R alleles, 0.49, right, 49%. Whoops. All right, what's the proportion of little r alleles? So we come over here, little r alleles, 0.51, right? 0.51, 51% basically. What's the proportion of homozygous recessive? Little r, right? What's the proportion of whitefish? Little r, 0.29, 29%. What's the proportion of heterozygotes, which are the spotted fish? We come over here. Big R, little r, heterozygote, 43%, 0.43. And then finally, what's the proportion of homozygous dominant, right? What proportion of all those fish are orange? Excuse me. And again, we go here, proportion 0.28, 28%. So at this point, you are done collecting data and you're done running the experiment for experiment 1, right? Experiment is done for experiment 1. The data is collected. Now at this point, you need to calculate some data. Now what you're really after is the percent change, right? This is really what we want to know. We want to know, yeah, kind of, I guess we could look at how the population size changed, but more what we're interested in is how did the alleles change? How did the genotypes, right? Do we have more orange fish? Do we have more white fish? Do we have more spotted fish? This is really what we're after. But to calculate this, the percent change, first we have to calculate this. We have to calculate the difference. Now, before I show you how to do it, I should point out, let me scroll up. There are written instructions right here. So everything I'm about to tell you is written down. Some people learn differently. If it helps you to read it, then there it is. If it helps you to watch your video or watch the video, then here we go. So here we go. Let's calculate the difference. So let's start with population size. We started with 196 and ended with 199, right? So the formula, let me show you the calculator, is you start with the number, excuse me, the first number you put in is the number you ended with, right? And that is 199. That is our final number, 199. And then you subtract your initial number. So minus, right? Hit the minus button right there, minus the original number, which is 196. And then the answer is 3, right? So the difference is 3, which makes sense because we started at 196, we ended with 199. That means, and the difference is plus 3. That means we went from 196 and we added 3 to get 199. That's what that means. Let's look at this. Again, we'll calculate it. We ended with 0.49. We subtract the number we started with, which is 52, or excuse me, 0.52. And the answer is negative 0.3. So right there, I put negative 0.3 because, oops, negative 0.3, because we went from 0.52 to 0.49, right? What's the difference in that? Well, it's a negative 0.3. Again, we'll do it again. I'll do less explanation because I've done 2 explanations already. So again, calculate. We ended with 0.51. We subtract 0.48. And we end up with positive 0.03. Positive 0.03. On this next one, again, same thing. Calculate. We ended with 0.29, so 0.29 minus what we started with, which was 0.24. And the answer is 0.05. So 0.05, a positive 0.05. One more time. We ended with 0.43. We subtract the number we started from, which is 0.49. And the answer is negative 0.06. Negative 0.06. And then finally, the last one to calculate is the difference in the big R genotype. Again, we started with 0.28, or excuse me, we ended with 0.28. We subtract the number we started from, which is 0.27. And what is the answer? Positive 0.01, 0.01. So there we go. We're done now calculating the difference. And let me point this out before I move forward. When I'm grading this, I don't have the time to do all your calculations and make sure every one of them is exactly correct. So your biggest thing, the first thing you should look for is make sure you have the proper sign. 196 to 199, that's an increase, so it should be positive. 0.52 to 0.49, that's a decrease, so it should be negative. 0.48 to 0.51, that's an increase, so it should be positive, right? These are the things I'm going to look for. Also, another thing you should keep in mind, hopefully this will alleviate some of your stress. Let's say, all right, here's the correct answer, right? 0.05 is the correct number for this. Now let's assume you messed up and put 0.04. And again, I'm not checking each one of your equations, right? I'm not going to check each one. So this, you might get away with this. This just probably won't jump off the page and say, oh, that's wrong. So a small mistake like that, you'll be okay. Again, let me write the correct answer. Here's the correct answer. However, if you do something wrong, and this, I see this a lot, people mess this up a lot. They mess up decimal points, right? So the proper answer is 0.05. If your answer is 0.5, that will jump out. Like I will notice that, right? That's a big difference, and you'll probably get it wrong. So be careful of signs. Be careful of decimal points. That's basically what I'm getting at. Moving forward. So again, we are done calculating the difference. And again, what we're mostly after is the percent change. And now that we have the difference, we can calculate the percent change. And again, to remind you, all these instructions are written right here, right? So if the video doesn't do it for you and you need to see written instructions, they are included in this lab. But here we go. To calculate the percent change, what we do is we take the difference and we divide it by the initial number. Let me give you an example. If you weighed 100 pounds in January and you weigh 150 pounds now, right? You could say, well, that person gained 50% weight, right? Because if you started at 100, what's 50% of 100? 50, right? So if you add that to 100, you end up with 150. Keep that in the back of your mind as I use these actually more specific numbers. So here we go. It's going to, again, the difference divided by the initial. So in this case, the difference is 3 divided by what we started with, which was 196. And that gives us our first number, but then technically to make it a percentage, we need to multiply it by 100 or move the decimal points twice to the right, either way. So the answer to that one is 1.53, right? You can see all that. But let's keep it simple and let's round. Let's do some rounding. So let's call that 2%. So there we go, 2%. All right, let's calculate this one. Now the calculator, we start, or the difference is negative 0.03. So negative, oops, excuse me, let me start that one over. Calculate, there we go. Negative 0.03. We're going to divide that by the initial number, which was 0.52. We get our number, and again, we have to multiply it by 100 to turn it into a percentage. And then it disappeared. Let's do it again. I'm gonna do it quickly since you've seen how I did it. But the difference divided by the initial times 100. All right, and the answer is negative 5 point blah blah blah. We're gonna round that up to -6. So -6. Same thing here. Let's calculate this. We're trying to do this one. So we have the difference, which is 0.03 divided by the initial 0.48. Get that number, multiply it by 100. And the answer is 6.25. We're going to round that down to 6%. All right. Here we go again. The difference is 0.05. We're going to divide that by the initial number, which is 0.24. Take that, multiply it by 100 to make it a percentage. And the answer is 20.83, so on and so forth. We're going to round that up to 21%. 21%. All right, almost done. We have negative 0.06 divided by 0.49. Multiply that by 100. And the answer is, let's round that down to negative 12%. Negative 12. We come here. Again, calculate. We have the difference. So 0.01 divided by the initial, which is 0.27. We get our number. We multiply it by 100 to make it a percentage. And the answer is rounded up to 4%. So at this point, we are done with experiment 4. We ran the experiment, we collected the data, and again, that was this right here. This is the data we collected. And then we've calculated some numbers based off of the data that we recorded. We are done with the experiment of experiment 1. And this is a lot of numbers and it's a lot of steps, and I don't want you to get caught up in putting in numbers into boxes and making sure your math is correct, because this is not a math class. I want you to understand what this means. And I talk about this later and in a different video, but I want to go ahead and touch on it now. What we're saying here is the population size grew by 2%. Oops, excuse me, let me fix this. And we're saying that the proportion of big R's in the whole gene pool went down by 6%, which makes sense because obviously the proportion of little r's went up by 6%. We also found out that the proportion of little r, right, the little r, the homozygous recessive, the white fish, that number increased by 21%. The proportion of the gold, the spotted fish, right, the heterozygotes, the big R, little r, it went down 12%. The proportion of homozygous dominant, proportion of big R, the proportion of orange fish, it went up 4%. That's what all these numbers mean. And again, if you're so focused on the numbers and you don't actually understand what it means, it's completely useless. So when you're done, just kind of take a look at your information and see what you have figured out. Because again, the whole purpose of the experiment is to test your hypothesis. So that is the end of how to do experiment 1. There will be other videos to show you how to do the other experiments. But if you feel confident after this video, you don't necessarily need to watch the other ones, because the other ones kind of go over what I've already gone over, just in a different context. So that's that. Let me go back over here, and we will stop the video.